Imagine a world where animals are specifically designed to combat human diseases. This isn't science fiction, it's the reality of transgenic animals. These remarkable creatures carry genes from other species, inserted through the marvel of genetic engineering. This field allows us to manipulate the building blocks of life itself. DNA. Picture DNA as a long, winding ladder. Each rung on this ladder is made up of two chemicals called bases. There are 4 different bases, represented by the letters A, T, C, and G. These bases pair up in a specific way, A with T and C with G. The order of these base pairs determines the genetic code, similar to how the order of letters forms words and sentences. Genetic engineering allows us to cut and paste sections of this DNA ladder. We can take a gene with a desirable trait from one organism and insert it into the DNA of another. This process, called transgenesis, is like adding a new word to a sentence, altering its meaning. The creation of transgenic animals started in the 1970s. Scientists were able to insert foreign DNA into mouse embryos, leading to the birth of the first transgenic animals. This breakthrough opened up a world of possibilities in various fields, from medicine to agriculture. Creating a transgenic animal is a complex and delicate process. Several techniques have been developed over the years, each with its own advantages and limitations. One common method is called pro-nuclear microinjection. This technique involves injecting the desired gene into a fertilized egg before the DNA of the sperm and egg have fused. The egg is then implanted into a surrogate mother, and if successful, the resulting offspring will carry the foreign gene in all its cells. Another technique utilizes viruses as vectors to deliver the desired gene into the host's genome. Viruses are essentially packages of genetic material that have evolved to efficiently insert their DNA into host cells. Scientists can modify these viruses, removing their harmful components and replacing them with the desired gene. A more recent technique called CRISPR-Cas9 has revolutionized genetic engineering. CRISPR-Cas9 acts like a pair of molecular scissors, allowing scientists to cut and paste DNA with unprecedented precision. This technique has made it faster, easier, and more affordable to create transgenic animals. Regardless of the technique used, the creation of transgenic animals requires rigorous testing and screening to ensure that the inserted gene is functioning correctly and that the animal's overall health and well-being are not compromised. Transgenic animals have become invaluable tools in biomedical research, offering insights into human diseases and paving the way for new treatments. One prominent example is the development of knockout mice. These mice have specific genes knocked out or inactivated, allowing scientists to study the role of these genes in various diseases. For instance, knockout mice have been instrumental in understanding and developing treatments for cancer, Alzheimer's disease, and cystic fibrosis. Transgenic animals are also used to produce therapeutic proteins, such as antibodies and hormones, in their milk or blood. These bioreactors provide a cost-effective and efficient way to manufacture complex proteins that would be difficult or expensive to produce synthetically. In agriculture, transgenic animals offer solutions to improve productivity, enhance animal welfare, and reduce the environmental impact of livestock farming. For example, scientists have developed transgenic salmon that grow much faster than their conventional counterparts, addressing the increasing demand for seafood while reducing pressure on wild fish populations. The ability to manipulate the genetic makeup of animals raises significant ethical concerns. Critics argue that creating transgenic animals is unnatural and could have unforeseen consequences for animal welfare and the environment. For example, the release of genetically modified animals into the wild could disrupt ecosystems and lead to the loss of biodiversity. There are also concerns about the potential for animal suffering, particularly if transgenic animals are created with genetic modifications that cause pain or distress. The use of animals in research is already a sensitive topic, and the creation of transgenic animals adds another layer of complexity to the debate. Despite these concerns, the potential benefits of transgenic animal research are undeniable. These animals offer a powerful tool for understanding and treating human diseases, improving agriculture, and addressing global challenges such as food security and climate change. The future of transgenic animals lies in finding a balance between scientific progress and ethical responsibility. Strict regulations and guidelines are essential to ensure that transgenic animals are used responsibly and humanely, while continued research and development will lead to new applications and innovations in this rapidly evolving field.